Hello out there, I am Rachel Easton from Harbor WildWatch, and we're ready to do a little experiment here to help people understand more about estuaries and some of the properties that different estuaries have. So thanks for tuning in. I am broadcasting from the Purdy Spit, so I do apologize if there's a bit of road noise or wind. So we're gonna, we can do this experiment, and I have to admit I've done of a whoops moment. I'm not a very good scientist, and I did not label my water samples. I have 4, and our job is going to be to try to figure out where these water samples came from. Now, luckily, I do remember where I got the water samples. The first is just around the corner from here, a place called Burley Creek. It's a freshwater. Went in, grabbed my bottle of water, chucked it in there, drove to my next location, which was right here at the Purdy Spit. Henderson Bay is the body of water that's right behind me. So we've got Burley Creek and Henderson Bay. Then this is the really fun part. I got to go in a kayak. I got to paddle out to underneath the Narrows Bridge, and let me tell you, that current just rips through there. It's a really, really fast place. Kind of an exciting and dangerous place to kayak, but I got a water sample there, so sort of right in the middle of Puget Sound. And then this part was really great. I drove a ways, all the way to the west coast and got a sample from the Pacific Ocean. So I have 4 water samples, but I didn't label them, which is a big problem. Now my water samples are useless. I can't do any testing with them. I can't do anything until I figure out which one came from where. Now, luckily, scientific instruments, things that scientists use pretty regularly. You can use similarly shaped objects at home, right? You could use maybe a bud case, a little narrow container. You can use an eyedropper instead of a pipette. But I've got my different water samples, and what I'm going to do is show you how to stack water, because the water that's from the Pacific Ocean, the Puget Sound, and Henderson Bay all has salt in it. It has a different density than the fresh water in Burley Creek. So we can do some testing with these different colors and water samples to try to figure out which one came from where. So I'm going to take my pipette and my water sample, and we're going to start with red because why not? It's beautiful. And the pipette works like this. I kind of squish it from the air out. You can see my fingers are pushed together. And then I'm going to put it underwater, and it just slurps it right up. And I'm going to put that in 1 full squirt. So we've got a nice red water sample. Now, if I do the same thing with a different water sample, let's say I want to test green next, get another squirt and put it in there. Uh-oh. I don't see any layering at all. No stacking. I just got kind of brownish water, which is not super helpful. So I'm going to dump that out. I've got a little extra cup here to catch it and start with a clean. And we'll go with it again. Why not? And I'll put one there. The trick is on the second sample to add it slowly. So I'm going to add a squirt of green, and the only way that I can think to slow down gravity is to tilt my test tube. And I'm actually going to tilt it pretty far. So we'll slowly add this, trying not to mix it too much. And then when I tilt it up, what we should see is one color on top and one color on the bottom. The color on the bottom is more dense. It has more salt in it. So right now, doing this first experiment, the only thing that I know is that red could not have come from the creek, because fresh water would not be on the bottom. It doesn't have the salt in it, or the fresh water doesn't have the salt, so the salt makes it more dense and heavier on the bottom here. So let's do another test. Got another test tube here. Let's do red again and see if we can figure out some more by process of elimination. So I've got this red color right here, and I'm going to add, let's do blue. Why not? Nice, pretty blue. So I'm gonna again tilt my test tube to try to slow down this drop of water and just gently squeeze it through there. This is where it's kind of fun to make predictions about it. And you can say, wow, I think maybe I know a bit about red. Ooh, look at that. You see that? Great layering right there. So right here, you got blue and red again on the bottom. All we still know is that red has more salt in it. We still have to do a bit of testing in order to figure out the rest. So we still know red can't be Burley Creek. And red is heavier than 2 of our locations. So that might tell you a little bit about where we're coming. So we're gonna go with red one more time. Nice big squirt in there. And there's one last color to test of my 4. I've got yellow next. So maybe you want to make a prediction about whether yellow is going to be heavier or lighter. And we'll slowly and carefully add that color. All right, I think I'm seeing that yellow top there. So if red is the heaviest, the most in, now yellow and blue might make green, but that doesn't mean that it's the same as my green water sample. So nice layering there. We can clearly see the blue underneath. The blue is more dense. So blue's more dense than yellow and less dense than red. We're starting to piece together our mystery, aren't we? So I'm gonna set this one aside. Let's test blue with our other color. Let's test against green. So let's start with green, why not? We'll put green in there first, and we'll do a nice full squirt of blue. I'm gonna tilt my test tube and go slowly to slow it down and try to avoid any mixing. Now you might be thinking that whatever color I put in there first is the heaviest, because that's kind of the way that it's gone. Every time I added the first color, it ended up on the bottom. But not this time. I added green, and then I added that blue, and the blue still went to the bottom and gave us a beautiful line here. So now we're starting to figure out where these water samples came from. We have a couple more tests to do. What I find is really helpful is to write it down in a greater than less than fashion. So we know now that red is the heaviest. It's greater than, it's more dense than every other color. What we're trying to figure out now comes next in that. And we listed we saw that green was less dense than blue and yellow is less dense. I'm confident that blue must be Puget Sound. That's be the one. It's the next most dense water sample in the Pacific Ocean. So I think we've got 2 of them figured out, and there's one last test to do. We need to test yellow and green together and see if we can figure out which one is Burley Creek and which one is Henderson Bay. So we'll put our green in there, and then a nice slow squirt of yellow, tilted and slowed down. And when we're all done with this, we'll make a stack of all 4 colors to show how the idea of density and water stacking. Check it out. We've got beautiful green on the bottom, which means yellow must be Burley Creek. Green is Henderson Bay. Blue was Puget Sound. Red, Pacific Ocean. Let's do one final stack, and then we can talk about what makes estuaries so incredibly important and why this body of water right behind me is one of the coolest estuaries around. So we'll start with the most dense. We'll put that nice big squirt from the Pacific Ocean, the most dense, the most saltwater concentration in the bottom of our test tube. And then what comes next? Puget Sound, so we'll put our blue water sample in here. And again, I want to slow it down and keep it tilted. And I'm going to actually keep it tilted for the rest of these colors so that we can see the reveal at the end. So we've got our red, nice slow squirt of blue. Puget Sound, the green next. So we'll add that one. We're stacking water here, and the property of density is what allows us to do this. Ooh, it's starting to rain on me. Who would have guessed rain in April? They say April showers bring Mayflowers, and what do Mayflowers bring? Pilgrims. All right, let's add our last color. Our Burley Creek, the fresh water should have no salt in it and will be very, very light, less dense. All right, and then we'll stand it up and see if we've got a good stack of colors. Here we go. Check it out. We've got Pacific Ocean, Puget Sound, Henderson Bay, and then our fresh. This is the order that you came up with. All these different tests together. Oh, good job. Now, what's really cool about Estuary and where we live here in the Western Washington is that we're surrounded by this beautiful body of salt water. This is Henderson Bay. It's a part of Puget Sound and the Greater Salish Sea, and it is an estuary. And by definition, it's where fresh water and salt water mix together. And you might remember from our first video that Stina and I got really into mixing it together with our estuary dance, fresh water, salt water mixed together. Well, all of Puget Sound, because it has so many rivers and streams, over 10,000, that instead of having a spiraled shell on them, they have this one shell that's kind of shaped like a mountain. I see some barnacles, and these are live barnacles. You can tell that they're alive because there's a little trap door on the inside. Those two little doors are pieces of the barnacle shell that they pull closed so that they can withstand the low tide. There are some really, really cool little snails here. Check it out. We've got a periwinkle snail, and this one's called a checkered periwinkle. You can see those really pretty check marks on there. And these are all mollusks. They're shelled animals with a soft, fleshy body. The barnacles are in the arthropod group. They are related to crabs and shrimp. But we've got about a 0 foot tide today. So I'm gonna leave my water samples here and head down and see what we can see. Now I already see a couple animals. Maybe you can see them too. There are some clams that are squirting. So keep your eyes peeled. I'm gonna hope that I don't get squirted by any of these clams as we're walking along. And our tide has come up significantly since I got here. Our low tide was about at, I wanna say about 1:30 today. So I'm gonna make my way just to the edge of the shore here to see if we can see anything living, any interesting creatures around. Oh, this is called the rack line right here. Check it out. All this stuff just floating right in the water. This is going to get pushed by the high tide up onto the beach, and anything that's not anchored down, like this piece of eelgrass, is going to wash up and kind of be stranded up there. This is the only plant that lives in saltwater in Puget Sound. This is called eelgrass, Zostera marina, and check it out. There's some little teeny tiny eggs tucked inside here. Now, do I know what kind of eggs those are? No, I have no clue. All I know is lots and lots and lots of animals use the eelgrass habitat for laying eggs. In fact, just this week and for about the last month or so, throughout the Salish Sea, herring have been having a record year for spawning. And they've been laying eggs all over the eelgrass and the sargassum weed. They just were spawning all across Seattle, which was really, really neat to see. So let's take a look at, we're still in the middle intertidal zone. You can tell because of the green algae that's here. Now I had just right out there found some cool sand dollars, but I don't think they're going to be found this far up. But we can look and see. And what I usually like to do in this zone is to look for substrate. So look for something that's got maybe a little place to hide. Now this rock, I can tell is a rock that's been flipped because it should go this direction. Notice the algae's growing on this side. Well, algae can't grow under a rock. It needs the sunshine. So this is a rock. This should be the underside. This is a rock that got flipped. So we're going to return it the direction that it goes, being careful not to smush any little creatures underneath there. Oh, and I do see a little creature. Ooh, a very little creature. Can you see this little crab here on my fingers? This is a green shore crab. Green shore crabs are not always green, although this one happens to be. And they have a couple close cousins that live in this intertidal zone as well. There are purple shore crabs, and then juvenile of other species of crabs are pretty common to find here. Graceful crabs or red rock crabs. And you can always tell the green shore crabs, he's totally gone. Look how quickly that little guy disappeared. Excuse me. You can always tell because they have tiny, tiny hairs. Now here's an even smaller one. There's my finger for scale. So we're talking itty bitty little crab here. We'll let him go on his way. Let's check out another cool creature that I see. This is a juvenile heart cockle. And the heart cockles are a species of clam that doesn't really like to bury the way that other clams do. And you can see that heart shape right there. Go ahead and throw up a heart for the heart cockle. Isn't that beautiful? This is an animal that gets about the size of my fist, so nice and big, and they like to sit on the top, and when a predator like a sea star comes up to them to try to have a snack, they will kick their foot out and be able to, ooh, I just missed that big clam squirt. I don't know if you guys could hear it. It was a pretty big one. Oh, there's some more over there. Of course. Now it's really starting to rain on me. Let's see what else we see. The sea creatures here, they don't really mind the rain. Remember, they're estuary creatures. They're used to freshwater and saltwater mixing. So when the rain comes, they don't really mind all that much. So here's an oyster shell. It's a Pacific oyster. No oysters home here though. It's just the shell. Although the shell has become habitat for a little dove snail and a little blobby thing that I can't quite identify. Let's see. Let's look at some of these larger rocks. Now, rocks that have space underneath them tend to be places where you can see lots of animal life because the animals can get under there. So let me just gently roll this back and see if we can see anything alive. Another green shore crab. Another limpet. On the rock itself is another great place to look, and check it out. I can see all these tiny baby barnacles. All of these little tiny bumps that you see here are the next generation of barnacles that have settled on this rock. Now these barnacle shells right here are empty and there's nobody living there anymore. Those animals have died, probably because their rock maybe got oriented the wrong way, but lots and lots of new life has colonized this spot here. Everything really becomes habitat. So we'll roll that rock back in place. Let's take a look maybe over here and see what else we can see. So here's another rock. This one was flipped, and I can tell because there's this blank spot in the algae right here, and all these little baby barnacles would like to be down, and I bet if I roll this over, there'll be some, what do you know, algae on the top of that rock. So it's important to put rocks back the way that they are because the animals that settle on them have settled on that rock for a reason, and in that particular spot for a good reason. Cool to see. All right. Now, this green stuff that we're looking at is a really cool algae. Let's see if I can pull up a rock and show it to you. This is called ulva. And ulva takes kind of two forms. It makes these beautiful blades, or it kind of curls and looks more like noodles. Its common name is gutweed. So you can kind of see that intestine looking algae. There's some red and brown algae in here too. Oh, it's really raining, you guys. I don't know if you can see that big storm cloud is right over us. It is a wet and soggy day here at the beach, but we'll keep going. Why not? Oh, here, check this out. Okay, here is one of those creatures I was hoping to show you today. This is a sand dollar. This one is a live sand sand dollar, although on the beach you'll commonly see dead sand dollars. You can tell that it's alive because of the color and the texture of it. So live sand dollars are always kind of spiny, and sometimes they even move their spines when you flip them over. So hopefully you can see the little wiggling and scooching around of those tube feet. They have a mouth in the center, and when they're alive, they will not show you their open mouth. But a dead sand dollar doesn't care, and you'll see a hole right in the center. So if you flip a sand dollar over and the flat side has a hole, then you know it's a dead one. This one is alive, and I'm actually gonna tuck him back a little bit down away from sitting on the top of the algae. Here's another cool creature, another snail. So this snail, you can see his foot is what goes into the shell, and behind the foot they have this really cool operculum, this little trap door that closes that helps seal the snail inside there. I'll set him back down. Let's see what else we can find down here. I've got some brown algae. Nice. Some red algae. It's that filamentous red algae. Really, really pretty when it's pressed and flattened on a piece of canvas or paper. Oh, all right. I think I am getting soaked out here. It might be time to call it a day, but we got a couple more low tides coming up this week, and in about 2 weeks from now we have some really, really super low tides, and we're going to be broadcasting live from different beaches. We've got some partnerships going with Tacoma Metro Parks to be on their beaches. We've got partnerships with PennMet Parks and our state parks to be able to bring you some really cool beach while we're all stuck at home. So thank you for tuning in today. We'd love any donation that you wanted to make to our organization to help us continue to provide these free resources. Oh my gosh, I am getting so soaked out here. Thank you all for tuning in and for watching. Oh, check it out. I don't know if you guys can see. There's a cool bird behind me. It's a Caspian tern. Usually they don't show up in our area around until summer, so I'm kind of intrigued why that guy's here now, but well, must be some good eating in the area. They come here to eat the herring. Okay, I'm gonna get dry and get off the beach. Thank you all for tuning in. We'll see you again next week.